biology with a minor in psychology from Juniata College. In 2019, Allison came to our group as an undergraduate research scholar under the Northeast Woody Biomass Warm Season Biomass Consortium. It was a big project, $10 million project with the USDA. And she came to work in the SkyCap project. In that project, she surveyed pests and diseases on willow. And after doing that, going up and down the rows of willow, she learned how to fly a drone. And then she used the drone to do that. Not only did she learn how to fly a drone, but she obtained her drone pilot license. So she's a licensed pilot for drones. During the summer of 2020, Allison joined NASA Jet Propulsion Laboratory in California, Pasadena, California, as an undergraduate research intern. And after finishing her undergraduate intern at California, she joined back our group to do her master's in plant science. Allison will share with us the research work she has been doing over the past year and a half. I ask please to put the questions in the chat or keep them in mind. And after she finishes, it will have some time for questions. So Allison, it's my great pleasure to give you the floor. Please take it.
Thank you, Felipe. Okay, hello everyone. My name is Koehle, and I am a master's student in the Department of Plant Science, and I'm going to be presenting to you my research on smart tillage and the regulation of nitrous oxide emissions in organic agriculture. So first, I'm going to give a little background on nitrous oxide emissions broadly, and then specifically within agriculture, and then even more specifically within organic agriculture. And then I will talk a little bit about the new method of measuring greenhouse gas emissions that we created as part of this research. And then lastly, the part of my research which deals with smart tillage and using it to regulate nitrous oxide emissions in organic systems. So nitrous oxide is a potent greenhouse gas. It has a global warming potential that is about 300 times that of CO2, meaning that it is about 300 times as powerful at warming our atmosphere as CO2 is. Of all greenhouse gas emissions, nitrous oxide accounts for about 10% of the total, but within that 10%, agriculture claims about 78% of that. And this is mainly due to soil management and the over application of nitrogen fertilizers. Nitrous oxide is emitted through two main processes. The first is through nitrification, in which we are converting ammonium to nitrate. And we see nitrous oxide as a byproduct of this conversion and is specifically emitted in environments of high oxygen. And this form of nitrous oxide that we get from nitrification produces a steadier background rate. So it doesn't emit the bulk of emissions. This is what denitrification does. So denitrification is the second process that forms nitrous oxide. And this is the process of converting nitrate to dinitrogen gas, and it has nitrous oxide as an intermediate. There are specific conditions in which we see high emissions of nitrous oxide, and that is when we have an increase in overall mineral nitrogen supply within the soil. This overall increases the rate of denitrification, and then as a result of that, it overall increases the amount of nitrous oxide that is emitted. And additionally, if we have an increasing supply of decomposable carbon in the soil, we have an increasing rate of soil respiration within the, within the soil layer. And with an increasing amount of soil respiration, we are consuming more oxygen within the soil and decreasing the amount that can be used to fully reduce nitrous oxide to dinitrogen gas. And this forms a type of incomplete denitrification, where the denitrification process ends at nitrous oxide and we see high emissions. Specifically within organic agriculture, we see increased nitrous oxide emissions when we are incorporating fertility inputs. This is specifically cover crop and manure residue. We see hot moments of episodic emissions from nitrous oxide during specific moments, including cover crop termination and manure application. These two incidences increase the overall mineral nitrogen supply within the soil, which increases the overall amount of denitrification, which I had explained previously. Tillage aerates the soil, increasing overall soil respiration and decreasing oxygen supply, resulting in increased nitrous oxide emissions. And then precipitation, lastly, it decreases the overall oxygen supply within the soil because precipitation moistens the soil, increases the water filled pore space, and which provides a lesser chance of oxygen to be able to get to nitrous oxide in order to fully reduce it to direct nitrogen gas. So next, I'm going to be talking about the semi-automatic method of sampling greenhouse gas emissions that we used in the field this summer. So as I mentioned previously, during these hot moments of nitrous oxide emissions, we see peak emissions. And it's important for us to be able to measure nitrous oxide during these peak emissions in order to accurately measure the amount of emissions that we're getting within a system. And this being able to measure nitrous oxide or greenhouse gas emissions more broadly is used or is done using non-steady state or static chambers. Essentially how this works, I will switch to the pointer if I can, here we go. So essentially how this works, this is the static chamber in this figure right here. We place that on top of the soil and we allow for greenhouse gas emissions from the soil to accumulate within the chamber's headspace here. And then we sample at specific intervals from the chamber headspace through these two ports here. And then by sampling from the chamber headspace, we are able to gather the change in greenhouse gas emissions through whatever amount of time that we have the chamber placed on the ground. And then this change in greenhouse gas emissions within the chamber over time is what our emission calculation is. So there's two ways in which we can sample from chamber headspace. The first is manually sampling, which is when operators, like I said, manually sample from the chamber headspace. This is what we see in this top figure here. So we are manually sampling from chamber headspace, and this is a relatively inexpensive way of measuring greenhouse gas emissions, but it is also very time and energy intensive, especially if you have a lot of treatments and your plots are far from each other. It can get very time consuming and taxing to the body. And then on the opposite end of that, we have the automatic sampling, in which we have technology that automatically samples from the chamber headspace. This is a more expensive version, and it can be a little less practical depending on how many plots you need to sample and being able to transport the automatic sampling system from one plot to the next. So the objective of this research was to develop and test a semi-automated gas sampling system that provides the sampling efficiency of the automated chamber, but at a cost more comparable to that of the manual chamber. Oh no, here, I'll try this, there we go. So this is the semi-automatic sampling system here. So we have 5 main portions of this sampling system. So the first is the power source, which is located right here. So this is the battery which is supplying power to the whole system. And then we have the air pump, which is located right here. Essentially how this works is that the pump is drawing air from the port right here that's inside the chamber headspace. So it draws air, the gas from within the chamber headspace through this tubing, through this manifold, and then through the pump and then back into the chamber. And this essentially provides a continuous mixing of headspace air so that we're taking a representative sample of the concentration that's within the chamber headspace. And then we have the controller, which is located right here. The controller is what we are programming in order to take a sample at specific times using these valves right here. So the controller is able to tell each of the valves to open at a specific time, stay open for a specific amount of time, and then close. And then the sample is being taken through this spring-loaded syringe mechanism, in which the spring located right here is providing the tension needed in order to draw a sample from the chamber headspace. And I'll have a better visual of how this works in this next slide. I may need to go to the pointer. I don't know how to get there though. How exactly do I? Oh, there we go. Okay, so this is the semi-automatic sampling protocol. So this is a soil collar, which we want to place in the soil at least one day prior to our sampling. And this provides a way of sealing off the headspace atmosphere from the outside atmosphere. So we place the chamber on top of the soil collar, and then we start the controller, and this is signaling to start the sampling period. And then we seal the chamber top from the chamber base using these binder clips. And I'm not sure if you can hear it here. So that's the pump working right now. So when there's an auditory cue when starting the controller. So when we hear this pump going, we know that the chamber is working. So then once we have the chamber on the collar, we place each of these syringes. The default setting for each of these valves is to be closed. So by twisting on or connecting these fully dispensed syringes and then pulling the spring back, the syringe acts in a vacuum and prevents the plunger from drawing a sample. And then, so that's how, and then here is kind of a truncated version of the whole sampling process. So we can see I set it for a specific time to open each valve. So there's one, and look specifically right here, I guess. And then right after the sample is taken, these valves are closed so that there's no mixing of the sample that's being taken within the syringe with the rest of the headspace that's mixing. And then once all of the samples are collected within the syringes, we transfer the samples that are collected into pre-evacuated vials. And this is sped up a little bit just for time's sake. Not actually that quick. So we unhook we unscrew each of the syringes from the valves. We place the needle onto the top as soon as we take it off, and then we undo the spring, and then that allows us to be able to put the samples into our vials. So then once all of the samples are transferred into the pre-evacuated vials, we just clean up the system, and then we can move on to the next plot that we have to sample. So in order to test these semi-automatic chambers against the conventionally used manual chambers, we conducted some tests at a fallow plot in the cover crop cocktails experiment. We used 10 chamber collars, or we pre-installed 10 chamber collars, which is what you can see in this image right here. We used 5 semi-automatic and 5 manual chambers, and we randomly assigned those to the 10 chamber collars that we pre-installed. We sampled simultaneously for 30 minutes, taking samples every 10 minutes. And then once one sampling period was finished, we switched the order. So any of the chamber or any of the collars that were previously assigned to be manual became semi-automatic and vice versa. And then we repeated the sampling period again. And then from there, we took our gas samples, we analyzed them through gas chromatography, and then we were able to calculate the emission as the change in greenhouse gas emission over time within each of the chambers. Yeah. So based on the results from the ANOVA comparing the carbon dioxide emissions gathered from the manual versus the semi-automatic, we found that there was no influence in the measured respiration rate. So essentially the manual and semi-automatic chambers were measuring similarly, or were measuring similar respiration rates on the days that we sampled. So here is a graph comparing the CO2 flux from each of the chambers. So on the x-axis is the semi-automatic with the manual on the y-axis. And then we also have a one-to-one line that's going through the graph. So essentially what the one-to-one line is providing is a way of comparing the two. So any samples that are closer to this one-to-one line are going to be more similar. Based on the distribution shown in this graph, we can see that it may be hinting toward automatic, showing a slightly higher emission rate compared to the manual because we have more samples on this side of the one-to-one line. This could potentially be due to the increased mixing of headspace air. So with the manual, you are only able to mix manually within the chamber headspace, and it's only used with a needle. Whereas for the semi-automatic, we are providing this continuous circulation the entire time we're sampling. But again, the ANOVA reveals that there's no difference in means. So for the purposes of this research, we are seeing, or they're measuring similarly. And then as for the specific respiration rates that were recorded from these chambers, the overall rate was low of CO2, about 9 kilograms of CO2 per hectare per day. So it would be important for us to be able to compare the manual and semi-automatic at higher flux rates as well, in addition to the lower ones that we used in this sample. And then some considerations and troubleshooting that we had to deal with using these throughout the seasons is weathering central PA summer conditions. So on particularly hot days and ones that were very sunny, we found that the heat and UV exposure degrades the plastic that the syringes are made out of. And we found that we had to replace those every 6 or 7 sampling days. Additionally, on particularly humid days, we found that when the syringes were fully dispensed, condensation would form within them, and that condensation would almost form like a suction between the plunger top and the top of the syringe, and it would prevent a sample from being drawn. So that was another thing that we had to note to make sure that the plunger was not fully touching the top of the syringe when we placed them onto the valves. And then lastly, there are electronic components of the system, so it's important not to use them in the rain. And then lastly, we had to complete pre-sampling inspections before every day of sampling. Because there are a lot of moving parts, we wanted to make sure that all the wires were connected in the correct place and that everything was working as it should be in order for the samples to effectively be drawn. So we did use these in the field in 2021, and it saved us a lot of time and energy. So opposed to using the manual chambers, we only had to go to the chambers and collect the chambers and set them up once. So just once to set them up and then once to collect the chambers. Whereas for the manual, we would have had to revisit the chamber about 4 times. So it limited the amount of soil disturbance as well. So because of this, we're going to continue using them in the coming field seasons. And then lastly, I was given the opportunity to present this research at the Tri-Society meeting this past November in Salt Lake City. And I got a lot of positive feedback, and a lot of people were really interested in the semi-automatic systems. That was great. So then the last portion of this presentation is dealing with the use of smart tillage and using it to regulate nitrous oxide emissions in organic systems. So based on preceding research completed by Saha et al., we found that jointly incorporating cover crop and manure residue increased nitrous oxide emissions. So this is the conventional way of managing cover crop and manure as fertility inputs. So this is a depiction of how it's used within the field. So we have our cover crop going before corn, which is to provide nutrients for corn growth. So we have our cover crop, we mow our cover crop so that the residue is placed on top of the soil. And then with the cover crop residue, we add manure, and then we invert the soil through moldboard plow, creating this layer of carbon and nitrogen rich nutrients or residue about 20 centimeters below the soil surface. And then we disc and plant. And this way of managing these fertility inputs causes microsite carbon and nitrogen saturation, which is one cause, or is a cause of high nitrous oxide emissions. So essentially, when we have this very concentrated layer of nutrients within the plow layer, we are increasing the amount of mineral nitrogen supply within that area, increasing the overall amount of denitrification, which increases nitrous oxide. We're also increasing the amount of decomposable carbon within that layer, which consumes oxygen and reduces oxygen supply within the soil, which causes incomplete denitrification and an increase in nitrous oxide emissions as well. This research by Saha et al. Also found that temporally decoupling manure and cover crop residue reduced peak nitrous oxide emissions. So what this means is that when we applied manure before cover crop planting, so right here, versus before corn planting right here, we were able to see a decrease in nitrous oxide emissions from 14 kilograms per hectare to 3.7 kilograms per hectare. So based on these findings, the question was raised, does spatially decoupling cover crop and manure residue within the plow layer decrease nitrous oxide emissions? So then, in addition, they found that legume cover crop biomass that exceeded a specific threshold increased nitrous oxide emissions. So based on these results, is it possible to decrease nitrous oxide emissions by removing above ground legume biomass and decreasing the overall mineral nitrogen supply that is provided through that above ground biomass? So the objectives of this research were to develop alternative techniques to managing fertility inputs, specifically cover crop and manure residue, with the aim of reducing nitrous oxide emissions and preserving nitrogen supply to crops. One of these alternative techniques is the use of smart tillage, which is a tillage technique that is aimed to regulate the distribution and concentration of cover crop and manure residue in the plow layer. And the aim of these smart tillage treatments is to be able to reduce that microsite nitrogen and carbon saturation that I previously mentioned by diluting and or secluding the cover or the organic residue within the plow layer. And then another alternative technique is to alter the cover crop composition and provide varying levels of nitrogen supply through cover crop biomass. So our first treatment is our cover crop treatments. We had 3 cover crop treatments: a crimson clover monoculture, a 3-species nitrogen-rich mixture, including field pea, crimson clover, and triticale. And then we had a triticale monoculture. And these 3 treatments are utilized to provide a gradient of nitrogen supply through both nitrogen uptake and nitrogen fixation. With the crimson clover supplying the highest amount of nitrogen, the 3-species mixture providing an intermediate range of nitrogen, and then the triticale providing the least amount of nitrogen. And then as for our smart tillage treatments, first we have our concentrated medley, which is just our business as usual, which I mentioned a couple slides back, in which we mow the cover crop so that the residue is on top of the soil, and then we add manure with our cover crop residue, and then we invert the soil through moldboard plow so that there's a concentrated layer of carbon and nitrogen rich residue within the plow layer, causing this microsite nitrogen and carbon saturation. So this is our control treatment, and then here's an image of what the field looked like before or after we plowed. The second treatment is the diluted secluded treatment, which essentially dilutes the cover crop residue in the top 10 centimeters of the soil layer before incorporating it into the soil. So in order to do this, we mow and disc the cover crop, and by disking, we are diluting the cover crop residue in the top 10 centimeters. And then we invert the soil through moldboard plow, add the manure to the top of the soil level or the soil layer, and then disc and plant. So by doing this treatment, we are providing a spatial separation with the manure residue on the top of the soil layer and the cover crop residue at the bottom of the plow layer. The second treatment is the secluded, concentrated secluded treatment, in which this is similar to the diluted secluded, but we are not diluting the cover crop residue in the top 10 centimeters of the soil layer before or incorporating. So essentially, we are mowing the cover crop so that the residue is on top of the plow, is on top of the soil layer, and then we invert the soil through moldboard plow so that the cover crop residue is now 20 centimeters below the soil surface. And then we add manure to the top of the soil layer, and then we disc and plant. Again, this provides a spatial separation between the manure residue at the top of the plow layer with the cover crop residue at the bottom. And then this image here gives kind of a visual depiction. I'm not sure. Yeah, you can't see it on the projector, but so here is where it's our diluted secluded treatment, where you can see that there's mild incorporation of our cover crop residue. And then right next to it is our secluded, concentrated secluded treatment, which we don't see the dilution of cover crops. So then our last smart tillage treatment is the biomass removal treatment, in which we are removing above ground biomass of crimson clover before incorporating. It follows similar field management to that of the control treatment, with the exception that we are removing the above ground biomass. So after the cover crop is mowed, then we remove the above ground biomass and add manure on top of the plow layer or on top of the soil layer. And then we invert the soil through moldboard plow, and then we disc and plant. So essentially what this does is it places the manure and root biomass residue about 20 centimeters below the soil surface, but it provides a lesser amount of nitrogen input. And with a lesser amount of mineral nitrogen input, we're hoping that this decreases nitrous oxide emissions. And then in these images here, this is an area, this is one of our study sites in which we removed the legume above ground biomass. This is a 2x4 meter section within our control treatment for the crimson clover plot. And within this area, it does look like there's still above ground biomass, and that's because there was volunteer wheat within each of our cover crop plots. So we only removed the above ground biomass. We did not remove the volunteer wheat biomass. And then here's just another depiction of how we marked the plots or we marked the biomass removal plot within the control plot. So this study was completed at the cover crop cocktails experiment. It follows a corn, soy, wheat rotation with the cover crop being tested here, being grown before corn. You can see here, this is all of our plots in red. So there are 3 tillage treatments, which we tested in each of the cover crop plots, each of the 3 cover crop treatments. And then we had 4 blocks total, which totals 36 plots. And then we had an additional 4 plots from the biomass removal treatment only within clover. So that totaled 40 plots that we were sampling. All of our plots were fertilized with poultry litter. And it's also important to note, like I mentioned on the previous slide, that there was volunteer wheat in all of our plots. It mostly impacted our crimson clover plot because that was to be a legume monoculture. In that plot, we had about 17% volunteer wheat biomass growing there. Nitrous oxide emissions were measured from June to October of 2021. We followed event-based sampling. We measured intensely around hot moments of peak nitrous oxide emissions. So in June, during the 1 month following cover crop and manure incorporation, we measured twice weekly. In July, we measured once weekly, and then thereafter we measured once every other week. And then we also sampled a day or two, or it depends, 2 or 3 days after rain events throughout the season. We used the semi-automatic sag chambers, which I discussed previously in the previous section of this presentation. We sampled every 15 minutes for 45 minutes, and we analyzed gas samples through gas chromatography. And then with results from the GC, we were able to calculate the change in greenhouse gas concentration over time, which is our emission. During the same day as nitrous oxide sampling, we measured soil temperature and water content. I gathered weather data, precipitation and air temperature from the USDA NRCS. And then I also analyzed soil mineral nitrogen. I took soil samples every other week for the first 8 weeks of the season, and then every 4 weeks thereafter. Using these soil samples, we calculated the gravimetric water content and the ammonium and nitrogen concentration. It's also important to note that I soil sampled in all of our treatments and all of our plots except for the biomass removal plot, because it was only about 2 by 4 meters. I didn't want to disturb the soil for when we were taking nitrous oxide samples. As for the corn harvest, we hand harvested a 2 meter by 2 row section within each plot. We dried the biomass for about 2 weeks at 60 degrees Celsius, and then we threshed the ears to gather the grain yield. As for data processing and analysis, I used random forest, which is a machine learning algorithm to test predictors of nitrous oxide emissions. The main predictors that we tested were carbon dioxide flux, cumulative precipitation 3 days prior to sampling, water-filled pore space, soil temperature, and nitrate and ammonium content. So random forest does give valuable information about the predictors of nitrous oxide emissions, but it doesn't give the statistical significance of the relationships between predictors in emissions. So I used multiple linear regression in order to test that. And then lastly, I used ANOVA to test the cover crop and smart tillage effects on nitrous oxide emissions. And then For our biomass removal treatments, I only compared our control treatment within crimson clover to our biomass removal treatment because both of those occurred within crimson clover. So based on the random forest results, we found that the random forest model that we used to untested predictors accounted for about 56% of the variability in nitrous oxide emissions. And then based on this graph here, this is the variable importance plot with percent increase in mean square error on the x-axis. And essentially an increase or higher, like the higher your percent increase in mean square error, the more important that variable is within the random forest model. So we have CO2 is our highest predictor with cumulative precipitation 3 days prior to sampling, which is right here, as our second highest, followed by nitrate, water-filled pore space, soil temperature, and then ammonium. And then as for our relationship between predictors and nitrous oxide emissions, there is a significant and positive relationship between all of them. The cumulative emissions within the system were high at about 10.2 kilograms of nitrous oxide per hectare per year. And a bulk of these emissions occurred following cover crop and manure incorporation. So on this graph here, we have month on the x-axis with each letter denoting the months of May, June, July, August, September. And then we have nitrous oxide flux on the y-axis. And this blue arrow right here indicates when cover crop and manure incorporation occurred. And we can see that the month or so following this event, we see very high nitrous oxide emissions. And during this time frame, we had average emissions ranging between 600 to 90 grams of nitrous oxide per hectare per day. As for our smart tillage treatments, there was no difference in nitrous oxide emissions between the treatments. Based on the averages, we can see that the control treatment had the lowest overall nitrous oxide emission and the diluted had the highest at 7.5 and 11.4 respectively. On this chart here, we see that the month is on the x-axis with nitrous oxide flux on the y, and there's a pretty even scatter from day to day of the emissions. So then also carbon dioxide flux did not differ between the three treatments, and then yield also did not differ between the three treatments. And then as for the biomass removal treatments, we also found no difference in carbon or in nitrous oxide flux between the two, with each measuring about 9 kilograms of nitrous oxide per hectare per year. Again, these were only in the crimson clover plots. As for the cover crop treatments, there was a significant difference in nitrous oxide emissions between treatments, with the legume cover crops emitting the highest, being the 3-species mixture in clover, measuring higher emissions compared to triticale. So triticale averaged about 6.5 kilograms per hectare per year, whereas the other two, the legume cover crops, averaged about 11. And then looking at this graph here, we have month on the x-axis, nitrous oxide flux on the y, and we can see that every day triticale in the green is emitting less than the other two treatments. Yield was also significantly different between the treatments, with clover emitting or with clover yielding the highest, and CO2 flux was not different between the three treatments. So overall, the cumulative nitrous oxide emissions within the system was high, with a peak or with a majority of these emissions occurring following cover crop and manure incorporation, which indicates that it is critical for us to be able to manage these organic residue inputs in order to reduce nitrous oxide emissions. There is a heavy importance of CO2 flux in the random forest model, which indicates that emissions may be from substrate-induced soil respiration. So when we are incorporating the carbon and nitrogen-rich cover crop and manure residue, we are increasing soil respiration, which decreases the overall oxygen supply within the soil and increases nitrous oxide emissions from there. And we can see a positive relationship between CO2 and nitrous oxide in these graphs here, with both being high following cover crop and manure incorporation, and then decreasing thereafter. There's also heavy importance of precipitation in the random forest model, which indicates that peak emissions may have also been from an increase in water-filled pore space due to rain and an overall low diffusivity of soil oxygen, which prevents the complete reduction of nitrous oxide to dinitrogen gas and releases higher amounts of nitrous oxide generally. And we can see this relationship in the graphs here, where we have these rain events occurring earlier in the season and coordinating with these peak nitrous oxide emissions. And then additionally, we have increases in water-filled pore space coordinating with these as well. But then from this graph, we can also see that there are a lot of high precipitation events later on in the season, which are not coordinating with peak fluxes in nitrous oxide. We see a bit of an increase here in coordination with these two peak events, but we do not see the peak emissions like we do in the beginning of the season. This is potentially due to the decreasing rate of nitrate within the soil throughout the season. So with a decreasing rate of, or with a decreasing concentration of nitrate within the soil, we're going to have an overall reduction in denitrification. So there isn't as much nitrate to be reduced within the denitrification process during that time, resulting in lower nitrous oxide emissions. So then the legumes we found emitted the highest carbon dioxide compared to triticale at 11 versus 6.5 kilograms of nitrous oxide per hectare per year. And this could potentially be due to differences in carbon to nitrogen ratio of the cover crop biomass. So the critical carbon to nitrogen ratio is between 26 to 29. So any residue that is above this amount is going to cause microbial immobilization and a reduction of the available nitrogen within the soil and an overall reduction in denitrification. And then any residue that is below this critical carbon to nitrogen ratio, we're going to have net nitrogen mineralization and an increasing supply of inorganic nitrogen within the soil. And then by comparing the carbon to nitrogen ratios, we can see that the legume biomass had below this critical value, whereas the triticale had above this critical value. So we would see an overall increase in nitrous oxide due to the increase in nitrogen within the legumes versus the higher carbon within the triticale, resulting in lower emissions. The removal of above ground legume biomass also did not decrease nitrous oxide emissions. This could potentially be due to the general overabundance of nitrogen within the system at the beginning of the season. So all of our plots were fertilized with poultry litter at about 180 kilograms of nitrogen per hectare. And this is a very readily available form of nitrogen within the soil compared to the cover crop above ground biomass. And the above ground biomass accounted for only about 30% of the nitrogen input, which does seem like a lot, but compared to the more readily available manure, the differences in nitrogen input may not have been enough to cause a reduction in nitrous oxide in that system. And then as for our smart tillage treatment, the diluted secluded and concentrated secluded did not decrease nitrous oxide emissions compared to our control treatment. And this could potentially be due to the fact that we did not allow enough distance. We did not allow enough spatial separation between our manure and cover crop residue within the plow layer. We didn't allow enough distance in order to eliminate that microsite nitrogen and carbon saturation. We potentially could have created an even thicker layer of carbon and nitrogen saturation within the plow layer. So by looking at these figures right here, these are the soil layers after manure and cover crop incorporation. So we see the manure on the top of the plow layer versus the diluted cover crop residue at the bottom, allowing about 5 centimeters of separation between the two. Whereas for the concentrated secluded, we have about 15 centimeters of separation between the two. So with, especially in the diluted, there isn't much space between the two. So after plowing, either soil compaction or rain can accelerate, or soil compaction can bring them closer, and then rain can accelerate leaching of nitrate within the soil layers, essentially taking the nitrate from the plow or from the manure residue layer and taking it down to the cover crop layer, and then not eliminating that microsite nitrogen carbon saturation. And then as for some considerations of this research, this is from only 1 year of data, so only 2021 field season. And we do plan to get one more year of data in the coming field seasons. Some future research could possibly be placing residue deep within the soil layer, specifically 30 centimeters, so lower than the incorporation depth of the control or of the business as usual treatments now. So placing the residue about 30 centimeters below within this figure. By placing them deeper within the soil layer, we can prevent fast decomposition because soil is colder, deeper within the plow layer. So by preventing fast decomposition, we can decrease the overall consumption of oxygen and potentially allow for more nitrous oxide to be fully reduced to dinitrogen gas and not be emitted. And then additionally, by placing the residue deeper within the soil layer, it provides a longer diffusion pathway for nitrous oxide. So nitrous oxide that is created in this layer here has to diffuse 30 centimeters through the plow layer in order to get to the atmosphere, which allows for more time in which nitrous oxide can come in contact with oxygen and be fully reduced to dinitrogen gas and not be emitted as that compound and emitted as dinitrogen instead. And then lastly, we can alter cover crop composition. So within this experiment, we found that triticale emitted the least, but it also yielded the least, indicating nitrogen stress. So it would be important to have a cover crop mixture which incorporates triticale, but also increases the amount of nitrogen that's being supplied, which is somewhere less than that was provided within this 3 species nitrogen mixture because we still had high emissions within that treatment. I want to thank Armin Kaminian, my advisor, for all of his guidance, my committee members, Jason and Liana, Felipe Montez for all the help in the field, Rosalie for all the help in the field, everyone in the committee and lab, especially Ellie for helping take nitrous oxide samples in the summer, everyone that works at the agronomy farm, and then Jen Harris as well for helping in the field this summer. Thank you.
Do we have time for some questions for Allison? Online question, Ellie. So what was the learning curve training like to use the static chamber? And are there other tools and further training that the next student could utilize to further the work you did?
So there wasn't much of a learning curve with the static chamber because I built them. So like I pretty much knew like how to use them because I built the process. But yeah, so there wasn't much of a learning curve there. As for expanding, it is a prototype. So there are a lot of issues that we need to work out before we use them in the field next year. We want to add some type of a shield to the top of the chamber in order to prevent the UV exposure affecting the plastic in the syringes. And then also the shield could help with rain events and kind of shielding the electronic systems from the rain. So there's some ways that we can expand it in that sense. Yeah, I think that's pretty much, I think anything, you can find improvements in anything if you think hard enough. So I'm sure that there are more minds working on it could definitely find some things that we could improve on it. Yeah.
Did you ever time yourself with the automated? Like I get that you have less trips.
Yeah. Yeah.
Time?
So we didn't time ourselves per se, but we did, like I did sample with the manual in the beginning of the season before we had the semi-automatic fully together. And so in total, during the sampling days, we had 40 plots to sample from the 4 blocks. So we had 10 per block. And in one sampling day, we were able to get through about 2.5 of the blocks with the manual chamber before it just got too late for us to sample. Whereas with the semi-automatic, for the rest of the season, we were able to finish all 4 blocks every single day that we went out unless like extenuating circumstances or anything. But so we didn't time ourselves, but we did, like I did experience both, like sampling with both, enough to provide anecdotal evidence that we know that they help. But yeah.
You mentioned that the N2O were high, so I was just kind of curious what like 10 kilograms per hectare of N2O emissions. You know, like in the implications or the greater scheme of greenhouse gas emissions, like what is total N2O emissions per hectare? You know,
Yeah.
now, and so how does that relate?
Yeah, so like the nitrous oxide goes into our atmosphere and it has a high residence time in the atmosphere. So the more that we're emitting, so think about, so 10 kilograms per hectare is equal to, I need to do quick math in my head. Like, I'm gonna get this wrong, I already know it. Just because I'm standing in front of everyone. So 10 times 300 is about 3,000. Yeah, okay. So we have 3,000, it's 3,000 carbon dioxide equivalents within 1 year, which is relatively high. So it's just kind of increasing, like with an increase in nitrous oxide in the atmosphere, we have a compound that has a higher residence time in the atmosphere. So it's going to stay in the atmosphere for longer, and it's going to kind of contribute more to the warming of our atmosphere because of that. So that's kind of the broader implications. Yeah, I don't know.
Just to follow up,
Yeah,
relative to other fuels,
yeah.
how high is our nitrous oxide compared to other fuels in the literature?
So because nitrous oxide is like one of the, or agriculture is one of the higher emissions, and within agriculture, we're applying so much nitrogen, we're inputting so much nitrogen in the system, it is much higher than a lot of other systems, I would say. Yeah.
Follow-up question on that. So
Okay.
what proportion of like the nitrogen applied in your research was like lost at?
So nitrogen applied, we had about, for each of the systems, it was about 271 was the total amount of nitrogen applied by, or within the clover, I should say. And by emitting about 10% or 10 kilograms per hectare as the average, well, actually, I can do this quick calculation in my head again. So clover had 11.1. So 11.1 grams of, or kilograms of nitrogen divided by 271 kilograms of nitrogen applied is somewhere around 5 or something, which is very similar to others reported in literature. So in literature, it's about 3 to 5% of the nitrogen that you apply is emitted as nitrous oxide. So that's similar to the rates that we're getting in other systems, similar in the higher range of those within other systems.
Go ahead.
Yeah, you can go back there first.
Yeah, just need to ask another question about the literature.
Yeah.
I just had a question of if there has ever been any kind of knowledge and kind of something akin to a lifetime analysis of nitric oxide, whether it gets into the atmosphere or not. In other words, over a longer term, it's like, what is the fate of nitrogen? Does it, and what percent will get found up in soil versus, in other words, if you were to follow this experiment out to 30, 40 years,
Yeah.
would you still see that nitrous oxide not reaching the atmosphere?
You mean from within the soil?
Yes. Like, what is, like, this is coming from a place of not really understanding nitrous oxide cycling. But well, bigger picture implications of this. Do you see in the literature indications that the fate of that nitrous oxide actually will not, and it's hard to, you can't track molecules, but do you see that will not actually enter the atmosphere? In In other words, can you correlate these kinds of findings to an actual long-term decrease in pollution?
So if I understand what you're saying, that are you asking that if we, so say in a perfect world, we're able to reduce nitrous oxide emissions in all agricultural systems, like will that impact the amount of nitrous oxide that's in the atmosphere?
Yeah, I guess what I'm kind of getting at is if we make management decisions that short term,
Yeah,
slow that, find it in the soil longer.
yeah. So
Does that pathway extend into the future? I mean, does it actually get found out into living?
I want to say that it will have an impact on the amount of nitrous oxide in the environment. Like if we are able to manage nitrous oxide, I mean, there has been a steady decrease in nitrous oxide emissions within agriculture in the past 30 or so years, a very little difference, like very little decrease. So I guess that's my answer. I'm not sure if that really does answer the question, but
I guess
okay. Okay.
long term.
Yeah,
So it has a half-life in the atmosphere of about 120 years.
Yeah. yeah.
So I think you said in the next steps that maybe 30-centimeter tillage depth is
Yeah.
needed. So I think that's probably maybe at least a 50% increase in the depth at which farmers would be using commercial tillage.
Yeah.
So I'm just wondering if you would gauge growers' willingness or receptiveness to going that much.
Yeah, so within the scope of the project, I only did like the field sampling stuff. I haven't really talked to a lot of farmers about the practices, so I can't really speak to that. I mean, I would hope that they would be interested in it. I don't
I
know.
can answer that.
Okay.
We have 2 farmers in New York that are they participated in the proposal, and they are strong contact. So we haven't presented this data to them. So we'll see. Yeah, we have farmers in the project. Part of the disconnect was COVID.
Yeah.
You know, they were engaged, we started, and then COVID came, and we haven't talked to them in 2 years, so we'll have to wait.
You can do Andrew and then Celeste.
On that same theme, I'm thinking about the order of tillage operations or the order of field operations in general and organic cropping systems. And I'm thinking about, the beautiful figures you showed where we have this, we're thinking about this layer of manure residue or cover crop residue in this nice compact layer within the soil profile. And I'm just wondering, in terms of future work, if it might be valuable, or maybe we've done it already, to sort of verify that, okay, we're inverting the soil profile and we're turning this residue in, but is it all down there in, at a nice 10 centimeter depth or 15 or 20 centimeter depth, or is there greater mixing that's occurring?
Yeah, so they actually did do that as part of the proposal. I don't have an image of it on here, but they dug to 20 centimeters and there's a very clear layer of cover crop residue.
I can take that too. Basically, part of the success of the proposal was we provided data for that. And the way we did it was we put sand color, the sand my daughter uses to play.
Colored sand.
Color sand,
Okay.
yes. Green or blue. And then immediately before the plow, the mow plow passes. And after passes, you see the sand in the bottom. I mean, it's incredible. The proposal has a picture I can send to you. It's incredible. I didn't expect that. But you see the sand completely, the layer of sand. So that's what the impetus for the proposal, and I think that was part of why we got funded.
And yeah, so maybe to John's point, I mean, certain producers are probably moldboard plowing to different depths.
Yeah.
I mean, and then some, I'm thinking from the Lancaster perspective, when the English plow, they're plowing deeper than when the Amish plow, tractor drawn plow versus horse drawn. So, there's some really aggressive moldboard plowing that occurs, and then there's some where, I'm not quite sure that we're getting complete inversion in all scenarios. And so I'm thinking about the implications of that.
Yeah.
So Armin has a comment about the conversation a few minutes ago. The 10 kilograms per hectare of nitrous oxide Allison measured is about 3 to 5 times higher or worse, which negates any optimistic soil carbon storage with no-till or perennial grass plantings. And then he said, yes, it matters. Are there any more questions?
Yes, so let's have
So
one.
can you go to the slide that it shows like the total?
Doesn't seem to want to work now. This,
Oh, yes,
this one?
the one, the next one, yes. So I'm interested in the data because I'm also working on my actual
Yeah.
data. So you have 1 kilogram of nitrous oxide nitrogen per hectare per year, right?
Yeah.
But you took measurement from June to October, like November. Did you collect it like throughout the year?
Yes, yeah, so I interpolated from the day we started sampling until the last day that we sampled, and I just used linear interpolation for that.
So do you think like for like a whole year, there would be like much more nitrous oxide?
So I don't think that there would be a little bit of an increase, but I don't think that it would be substantial, because a majority of the time, the rest of the time, there isn't going to be as high emissions as there is within the summer, like during the hotter months, or following when we incorporate those nitrogen and carbon sources. So a majority of the emissions, we don't see high emissions within the winter, or like in the colder months when the soil gets colder, or when it freezes. You don't see as much during that time.
So we need to wrap up here. One last question. All right, let's thank Allison.
I was wondering, given you might want to kill like super trees.
Yeah.
Nice
job, Al.
Thank you.
I was wondering, like,